and why we are doing biosensing, and what kind of antigens and pathogens we want to detect and all that. So that is a part of the video which I uploaded. So try to watch that. It may give you some ideas about what kind of topic you can choose. For example, it can help you because now we started getting into bioengineering or biotechnology, where we are integrating the engineering and technology aspects of the things. So these are important lectures, I think, for give you some ideas, some understanding. But that doesn't mean that you have to stick with these topics. This is just for guidelines so that you can have some background. So today we'll be covering basically polymerase chain reaction, and then we'll also cover, if we have time, some of the sequencing methods briefly, how the DNA is sequenced. And let's start with the DNA replication. So, all our genetic information is stored. So this will be a continuation of a previous lecture, which is lecture 15. So, and you might be confused why I'm starting with the replication right away, because lecture 15 will be ending at DNA replication. So, and there is some discussion about the genes and all that. So this will be continuation of it. Okay. So once you watch that, I think then things will be more in order. But anyways, I think we can. We don't need that background, by the way, to go through today's lecture. So it should be fine. So when all that in our nucleus, we have a DNA stored in the chromosomes, and that all the information is stored in the form of DNA. Okay. And so let's say if there is some gene, which can be a small segment of a DNA, sometimes a couple of different genes work together, and they express in terms of proteins. So let's say if there is a DNA inside a nucleus, how come that DNA information is transferred to ribosomes where the proteins is synthesized? So basically there are some messenger RNAs which are involved. And a messenger RNase actually comes to your double-strand DNA. So double-strand DNA unwinds, and then the information is copied to RNA. And once it is copied, then the RNA travels back to your ribosome, and then the information, with the help of other RNAs, there is a ribosomal RNA also involved in protein synthesis. And there is a transfer RNA also involved. So the point is the information is normally transferred from the code is in the DNA, and the DNA is replicated quite a few times when the cells are also divided, right? So the DNA also replicates. So for replication, why we are talking about the replication, because we will be discussing about the polymerase chain reaction, and there is also one homework about it. There will be homework 3, which we'll discuss in a minute, so make sure that you understand it. So first, for replication, let's say this is a segment we want to replicate. We have to first attach a primer with it. So primer is a small segment which we already know that will go and bind with that gene, some segment of the DNA. And then there is a DNA polymerase. It's an enzyme which actually polymerize or makes new DNA. So let's say if you have a single strand DNA and now your primer is attached, then polymerase comes there and start adding new bases. Based on if it's A, then it will add T. If it's G, it will add C. So in that way, it just keep adding on the bases, and then it elongates towards the 3' end. I told you, right? From 3', from 5', it doesn't elongate. And then in that way, fragments are. If there are more fragments, then in humans, in a cell, actually those fragments can also be attached with one another to make a full long RNA sequence, and that is done with the DNA ligase, where 2 DNA fragments can be combined with one another. And this is just an image to show you how the DNA polymerase looks like. So this is the DNA inside, and then this is how the polymerase is simulated, and it shows that it is actually comes and winds around the DNA and then start adding bases. Okay, there. Okay, so now let's make things simpler. So can we do the DNA amplification or DNA copying in our lab? That's the thing, and that's called the DNA amplification. So there are quite a few techniques which are used for DNA amplification, And the most common and most commonly used still in the labs and in the R&D and also disease diagnostics and in looking at different genes if they're expressed or not expressed. The most common technique is called polymerase chain reaction, and in short we call it a PCR. So in PCR, it's isothermal amplification. It's not isothermal amplification. We require a couple of different temperatures to complete a cycle of amplification. And I will go through those steps one by one, but you can have actually multiple copies of DNA made with this process, and it's very fast. So after each cycle, which can be about 1 minute long, you can have double the number of DNA which you want to replicate. Let's say if you start with 10, after one cycle, after 1 minute, you will have 20 copies. After that, the second cycle, another minute, you will have 40 copies. Then you will have 80 copies. So every time it gets doubled, okay? And it finds a tremendous amount of applications in cloning, sequencing, forensics, and a lot of other diagnosis applications for PCR. And Dr. Mullis, he got half of the Nobel Prize in 1993 for invention of PCR. So you can understand that it's a very important process. So the PCR itself, how it works. So in PCR, the double-stranded DNA is denatured by mild heating. So when we say denature, it means the double-stranded DNA naturally we consider that it's a double-stranded. It means two strands are complementary and they're already bound with one another. So we have to open up those double-stranded into a single strand, and that opening normally is done with the heating. So if you let's say heat up your DNA to 90-95 degrees centigrade, like boiling water, if you give that temperature, the DNA will open up and it will make a single strand very rapidly. In few seconds you can open up actually DNA, most of the DNAs. And so once that process is called denaturing of the DNA. And then once now your DNA is open, so now you have two copies, right? So let's look at the one copy only. So one copy of DNA which may have some sequence A, T, G, C, whatever. And then for the PCR, you need to know at the start that what kind of gene you're looking for. So means you need to know some, let's say for example, we want to test if someone has some infection. Let's take example of Zika. Okay, let's say if someone has a Zika virus in his or her blood, or Zika virus RNA. So Zika virus has a RNA in there. Okay, so and if we, in order to detect Zika virus, we can detect RNA. If we can detect RNA, then it means we can detect the disease, if that disease, the blood is infected or not. So people have already sequenced the Zika virus RNA. It means they already know the sequence of Zika virus RNA. So our target is known, like what is the sequence, that is known already. So it's not something unknown. If it's not unknown, then we cannot use a PCR. So we need to know what we are looking for. So let's say we know this is a sequence of a Zika virus RNA, then we design primers based on some segment of that sequence. The Zika virus RNA is very long. I think 7,000 bases or 10,000 bases pair long, so it's long. And so we don't want to amplify all of it. So we can amplify some segment of that RNA, right? Let's say if this much long, so we can just amplify some small segment of RNA, which is very specific for Zika virus. It has to be specific. Otherwise, let's say in a blood you have a DNA and RNA from a lot of other blood cells and body cells. So we have to choose a sequence which is only specific to Zika virus, because some sequences do repeat between different cells. So once you know that what kind of segment you want to look for Zika virus, and that is very specific for Zika virus, then we can just amplify that part of the RNA. And if you see that amplification, then it means the Zika virus was there. If there is no Zika virus, then you will not see the amplification. So basically, let's say this is a segment, you know the sequence, you put a primer there. So primer is this starting few bases, 20 bases let's say, which you know that it will go and bind with somewhere. And then polymerase comes, and then polymerase starts synthesizing more bases. So polymerase only works when you have a double-stranded, when your primers are attached with your single-strand DNA. If, let's say, you have a lot of DNAs, but primers don't attach with anything, then polymerase will not work. Okay, in order for polymerase to work, the primer has to come there as a starter. So to give indication to polymerase that, okay, come over here, let's start amplifying from this side. Let's start making a DNA from this side. Okay, so once you make the DNA, then we attach, once you have a DNA which is denatured, single-stranded, then you put a primer on one side, and then DNA polymerase actually start adding bases complementary to your single-strand DNA, and then in that way you can have from 2 single-strand DNA you have now 2 double-stranded DNA, right? And then if you denature that again, then you will have 4 single-strand DNA, right? And then again this whole process will continue. So you do it in cycles. Again you denature it, again you put primers which are already there in a solution. Again the polymerase comes there and start making a complementary of that, and then you open them again. So in that way you can have double number every time. So from single strand you make it actually double strand. So there are 3 temperatures which are involved in one cycle of amplification in a PCR. The first step, which is called the denaturization, we already discussed it. The DNA is heated for 97 degrees, at 97 degrees for a few seconds. That will denature the DNA. The second step is called annealing. Basically, to make it simple, annealing is when your primers, which you have designed, comes and bind with your single-strand DNA. So why we need to, and that annealing process is normally done at low temperature, about 50 to 60 degrees centigrade. Why we cannot do the annealing at 95 degrees centigrade? Why do you think? Because it's already at that temperature, DNA doesn't bind. So we have to bring it at low temperature so that the primers can bind. So once primer bind, then we change the temperature again to 72 degrees, 70 to 72 degrees. And that's called the elongation step, where the polymerase actually, this temperature is important for polymerase enzyme to work on your DNA and start building the bases. Polymerase might show some activity at 56 or maybe 60 degrees centigrade, but it works best at 70 degrees centigrade. And then your primers are added actually. So if you look at this structure.
In the previous one, when you said like the temperature in annealing at what temperature?
Annealing normally is 60 degrees, around 60 degrees. 60. 60.
Celsius.
Centigrade, yeah, degree centigrade. So in a PCR, and that changes a little bit. So, but in that range, from 55 to 60 degrees, because it depends on your primer sequence and the sequence which you are targeting. We'll not go into that discussion, but basically, if you know that some bases of DNA has A's and T's, right, and some have G's and C's, and G and C's have 3 hydrogen bonding, so they are more stronger, but the other ones are not that stronger. So if you have more A and T's, it means you might need more lower temperature for them to bind as compared to, so there are a few degrees differences, but in general it's in that range, 55 to 60 degrees. So in a PCR, this is another example, graphical way, and I'll show you also a brief video about 4 or 5 minutes to explain the process. I think then things will be more in order after watching the video. So let's say this is your target which you want to look at. So and you already know that they are ranged from 3' to 5', and from complementary, right? And then on the other side there is a 3' and a 5' on the other side. So it's an anti-parallel. And can someone tell me what that 5' means and 3' means?
Which carbon?
Which carbon is at that side? Yes. The phosphorus, which is at the backbone, it makes a bond between carbon 3 at one side and the carbon 5 on the other sugar group. So, and that makes a bridge between two bases. And when we are at when there's on the top, there is a C5 on the top. It means we are that is 5' direction. So normally it elongates towards the 3'. So let's say after denaturing step, you make it to single stranded, right? Blue hair, but you will also notice that the amplification would be towards the 3'N. Okay, in this case also it will be towards the 3'N. So we have now two sides, and then you have already primers which are designed. So basically, because those two double-stranded DNA, so this double-stranded DNA, they are anti-parallel. We normally have two primers. One is called the forward primer. One is they call the reverse primer. So one's amplify in one direction, one amplify in the other direction, and those primers will be different because it's complementary. So now those primers will go and bind with there, and then the polymerase will come and start amplifying a new DNA basis based on the sequence which is complementary to the primary sequence, and then now you have two double-stranded DNA after this. So the red one is a newly synthesized DNA. Then you do another cycle. So from here to here, one cycle is complete. So denaturization, annealing, elongation. Then denaturation again. So now you have 4 single-strand primers. Then denaturation, annealing. It shows in one step only. And then this is elongation. So now you have 4 single-strand DNA. And after that you have 8. Right? So every time it's getting double. So let's say how many DNA copies? If we start with the 1 DNA, double-strand DNA. After 10 cycles, how many DNA copies we will have? So how many single-strand DNAs we will have after 10 cycles? 2 to 10. Okay, and after 30 cycles, 2 to the power 30. 2 to the power 30 will be a big number. So it means if we start with even 1 DNA, we'll be ended up with the 2 to the power 30 copies in just 30 cycles. And that's amplification. And that's advantage. So it means even if there are a couple of viruses present in the blood, so you can amplify them and very rapidly in few minutes, not few minutes, but maybe within 1 hour, you can amplify them to a significant number so that they will be very visible. And that's the power of the technique. And now it can be any gene you want to look at. So let's say we want to look at if this person has that specific gene. It may be cancerous gene, it may be some other gene related with his color, or maybe his parents or whatever. So we want to look for some specific gene if it is present in his DNA or not. So once we have a DNA or RNA, then we can design our primers accordingly, right, which can target that segment. We can design actually primers which can target 20 different genes, okay, and we can look at 20 different genes, not at one time, but in a parallel direction. And where we see the amplification, then we know that this gene is present there. If there is no amplification, that gene is not. In that way, we can find out if that gene will be expressed, or at least that segment is there in the DNA or not. And then the whole field of actually diagnostics, not the whole, but most of the diagnostics, the standard method is a PCR so far. It has some limitations, which we'll talk about in a minute, but in general, this is a standard method. It doesn't matter, Ebola, Zika, HIV, you name it. Hepatitis, dengue, other flavors, all the infections, they can be sequenced, most of them can be sequenced with the PCR reaction. A lot of bacteria and fungi. So the only issue with the PCR is lab-based, so it doesn't work with the blood right away. That doesn't mean that if you take a blood, you put PCR primers there and it will start working. No. So the blood has a lot of other things there. For a PCR, that is debris. or I would say inhibitors for PCR amplification. So we have to first isolate the DNA or RNA which we want to amplify from the other debris. Let's say if it's a blood, what I will do is I will first centrifuge a blood to isolate plasma. Okay, so to remove the red blood cells from there, and WBC is only plasma which will have RNA or viruses, which viruses are small, for example. So then I will lyse those. Lyse means break them so that the DNA RNA can be freed into the solution. And once they're free, still there are a lot of inhibitors there. Still there are a lot of factors which actually eats away or degrade the DNA over a period of time. And so for that we have to optimize some conditions and add some reagents there at specific pH so that everything stays stable. And then we do the amplification. Then the amplification also, we need to use some equipment. It's called the thermocycler. So thermocycler is the equipment which is used to cycle the temperature, and it's expensive. Now you think about if you want to complete one cycle in 1 minute, for example, so you have to increase the temperature to 95, then bring it down to 60, then 72 in 1 minute, and the heat has to radiate quickly, right? And you have to increase the temperature, otherwise you will not be able to achieve it very faster. And that's why the thermocyclers are normally expensive. I don't know what technology they use for this purpose in the equipment, but they can quickly heat up the tubes, PCR tube, where you have the primers and the DNA very quickly. And that is fine. And we recently bought actually one PCR machine which can look at 4 different genes, or it has 4 different lasers. I'll tell you why we need lasers later on. But it costs about $37,000. I bought it last year for a lab. Okay, so if you maybe get the cheapest one, it may be, I don't know how much price would be, but it would be around 10 to 15K approximately. So that is fine in developed world, but it's not affordable in resource constrained settings. And let's say if you want to develop some point of care assay for some regions in a developing country, low income countries, or maybe in some village, or where we cannot have these fancy labs. So we need a technician to operate all these things. We also need some good reagents, so that we need an enzyme, we need a basis, we need a thermocycler. The equipment cost and then the material cost itself is expensive. Let's say if you want to get the primer synthesized, normally getting synthesized, you have to get it made from a company normally, or make it in a lab, but they are not very cheap. So that's why, and the price is definitely one problem. One PCR based detection, it costs about, these numbers can change, but it costs around $100. Okay, and $100 for one test is not affordable in many parts of the world. Even it's expensive here. If $100 I have to pay, it's expensive. And sometimes we have to do the detection and quantification of the viruses in diagnostics at least quite a few times. So then it becomes unaffordable for many people. So, and then the time it takes from first, it's not only one sample you are looking at, because you have one lab, one person can work on maybe a couple of samples at one time. But in a hospital setting, you might be having many people coming in, right, drawing blood, so you have to process all that. And there are some automated versions of a PCR where the the DNA isolation can be performed to some extent automatically, but again, you make it more automated, it will be more expensive. So the point is, and that's why there is always interest to look for other methods, how we can detect disease maybe rapidly, low cost. So these are different parameters which need improvement, like low cost, rapid, less labor intensive, and all that. You don't need to, if it is equipment free, that's ideal. Like, do you know any test which is done and which is equipment free? You don't need any equipment to read it. Do you know any test in your mind? How about the pregnancy test? That's equipment free, right? The pregnancy strips or ovulation kit, you don't need any equipment to do it. It just tells you whether it's a positive or negative, right? Just a one line. It automatically shows up. So that's an effort, just as an example to show that why we won't go for other technologies which are more affordable, more user-friendly, and equipment-free so that people can perform at home. But again, the standard for many DNA or nucleic acid-based assays or virus detection is PCR still. But there's again an effort to look for other methods so that we can make things cheaper, faster, less labor-intensive, and more accurate. But PCR is actually, in accuracy, is very good. It normally gives more than 90%, 95% sensitivity and specificity. Specificity is normally close to 100%. It's very specific if you design your primer in a good way. So let's watch this video about the PCR, then we'll continue with some other slides.
The polymerase chain reaction, or PCR, is an alternative to cloning for isolating a DNA sequence. PCR can selectively amplify a single copy of a desired sequence that is present in a complex mixture of other DNA molecules. In this example, a mixture of genomic DNA molecules, shown as blue ribbons, contains the sequence we wish to amplify, called the target sequence, shown in green. With PCR, we can make billions of copies of the target sequence without having to purify it beforehand. In the first cycle of PCR, the DNA molecules are melted by raising the temperature to 95 degrees. After the strands have separated, the temperature is lowered to 60 degrees so that a pair of short oligonucleotide primers, specific for the 3' ends of the target sequence, can bind, one primer on each strand. The primers then direct a DNA polymerase, called Taq, to synthesize the complementary DNA strands from free nucleotides. In the entire mixture of genomic DNA molecules, only DNA containing the target sequence is copied because the Taq polymerase can only copy molecules that have a primer attached. At the end of cycle 1, both strands of the genomic DNA molecule have been copied to form 2 partially double-stranded DNA molecules. In the next cycle, the same steps are repeated. DNA duplexes are again melted by heating to 95 degrees. Since Taq polymerase has the special property of being thermostable, it is not inactivated at this high temperature. The primers anneal to the target when the temperature is lowered, and the target strands are extended by the Taq polymerase. The temperature can be raised to 72 degrees to speed this reaction. At the end of cycle 2, 4 partially double-stranded molecules are produced. All contain the target sequence plus some adjacent DNA. During the third cycle, the DNA strands are again melted. The primers hybridize to the target sequence, and the Taq polymerase synthesizes complementary DNA strands. Cycle 3 produces 2 doubly stranded molecules that contain only the target sequence, as well as 6 longer molecules that contain the target and flanking sequence. In subsequent cycles, the number of target sequence molecules will increase exponentially. At cycle 4, the DNA strands are melted, the primers anneal, and the DNA strands are copied as in the previous cycles. Eight copies of the target molecule and 8 longer molecules are produced. In cycle 5, the heating annealing and synthesis steps are repeated. By the end of cycle 5, there are 22 target molecules and 10 longer ones. The number of target sequences continues to grow exponentially at each cycle. By the end of cycle 30, the target sequence has grown to more than 1 billion double-stranded target molecules, whereas there are only 60 copies of the longer DNA molecules. This means that the final product is virtually pure target sequence. Thus, like cloning, PCR selectively generates a large number of copies of a desired DNA sequence from the original mixture of DNA sequences.
Okay, so that's the PCR, and now let's say we have, let me see what slides we have. Okay. So now let's say we have now a PCR, how we can quantify that the gene which we're looking at is really amplified or not. Because you're amplifying it, but they're very small, right? The thickness of the DNA single strand is very, very thin. And how do you know that it is actually amplified? What do you think? How we can really look at that? Any ideas? What can be the possible way to amplify it? Oh, sorry, to see the amplification, whether it's amplified or not. So if it is present at a very high concentration, then the solution becomes a little bit clouded. Okay, so that's the one way, so that you can see. But then it has to be super high concentration that you will be able to see the DNA is there. It's a cloudy a bit. But that cloudiness can be from some other factors also. So that's not a best way to do it. So the other methods are, the other method is that we can use some dyes, some fluorescent dyes, which can bind with the DNA, double-strand DNA, right? And then we can see it under the microscope, fluorescent microscope, or with the laser light, whether that dye is shining or not. So there is one dye, it's called the SYBR staining, and there are quite a few other dyes available, but this is one option. And but let's before that, we can also run a gel, by the way, electrophoresis gel, where you once after the amplification. So let's look at first the gel. Let's say if we don't want to use a dye, for example, or we can use some dye which is not very specific, because if we use a dye which can bind with the DNA, so then there is a possibility there may be some other DNA present in the mixture, so then the dye will also attach with it. So the one way to actually separate the DNA based on its length, so let's say if we are amplifying a gene which is 500 base pair long, for example, and so what we can do is we can run a gel in the regular agarose gel, so where there is a gel here, and then let's say you apply a negative, this is positive, and then you put all the DNAs there. So DNA will move, travel based on their length. They will go to different places in the time. So let's say after 1 hour, so you will get some DNA altogether, which will be of same length. So there will be some DNA here, some DNA here, and maybe a lot of here. This will be most longest one. So the smallest one actually go further, and now you also have a we call it a DNA ladder. Okay, where we already know that what kind of lengths are present in the ladder. So we always run a ladder besides this gel. So at one panel, let's say here we run a ladder. So ladder, we have a lot of bands. You will see a lot of bands here, and let's say starting from 100 base pair, this may be 200, this may be 500, this may be 600, this may be 1 kilo base pair long, this may be 5 kilo base pair long. So the ladder, we already know that what is the length of the DNA in the ladder. So once you see the ladder, it can give you, it's kind of a scale. So you can then, now let's say if we are looking at 500, let's say, so what segment from here will be 500? Only this one, right? So this will be close to 500. So then we know that, okay, this segment which we are seeing there, it may be our gene which we were looking at, right? And that have amplified. So basically in reality, when you run this kind of assay, so you will see maybe some large DNA is there, and then you will see that segment there, okay, mostly. And that is your amplification product. And that you can stain it with any dye which binds with the nucleic acid. So if you see the color change there or the stain there, it means that segment, that length of segment has amplified, okay? But it doesn't tell you that whether it's the same sequence or not, but it tells you that that is amplified product. So we consider our actually PCR amplification works. So if it works, it means then that's why we have that amplification. But if PCR doesn't work, or if let's say the target is not there, in that case we should not be able to see that. We should not be seeing that band there. We can use a SYBR stain for staining. So the SYBR has a property that it binds very well. Let's say this is a double-stranded DNA. So it binds very well with the double-stranded DNA. So when you have a double-strand, so let's say if there is a RNA or single-stranded DNA present in the solution, so then SYBR will not bind with that. So in that way you can avoid some kind of non-specific binding. Okay, and then you can see this stain under the fluorescent microscopy. Look at it will shine very quickly. And you can also use this stain with the gel. So then the gel will also give you fluorescence. And there are some readers which can look at the gels on different wavelengths. Okay, so you shine the different wavelength of light. If the dye is giving fluorescence, it means it's there, it's attached there. So this is one kind of reaction. There are other techniques which are also used. For example, the one which is written in the slide is called TaqMan, Polymerase, or TaqMan PCR. So in the TaqMan, sometimes we have interest to see if our PCR is working or not. So for that, we want to see that after each reaction, we look at so that's where the new PCR equipments, which are a little bit latest ones, comes into play. So they already have lasers. So they keep shining laser on your sample and then keep detecting the fluorescence during the reaction. So as the product is getting amplified, you will see the increase in the fluorescence signal. So for that, TaqMan actually PCR, you can see quite a few kits which are available from the TaqMan company and PCR products. And so in this case, what they did, which was slightly different. So let's say if this is your DNA. Now you have your primer, right? Let's say this is a primer which is attached. This is normal PCR, and then your polymerase comes and start adding bases. So in the TaqMan, what you have actually another small primer which binds not at this place but some other place on the gene. Okay, and this primer has actually two dyes already attached with it, and one dye is called quencher, and this is called reporter. So when the quencher and reporter are present in close proximity, then what happens? You don't see any fluorescent signal. But when quencher and reporter are broken down and they go somewhere else, like far from each other, then you will see that reporter will start giving you fluorescence. So basically, this quencher is some material which can absorb that wavelength of light which the reporter is giving. So what happens as the DNA is getting synthesized, and when it sees this probe there, the polymerase actually, the TaqMan polymerase kicks that off and breaks it, and then starts synthesizing. So if it comes in a way, it's just push it away and then start making the bases as it was doing before. And then now these two, reporter and quencher, will be actually moving in a solution, and now the reporter can give you fluorescence. So in each cycle then you have more and more reporters available, and then you can see if your DNA is amplifying during the reaction or not. Okay, so and that kind of reaction, we call it as real-time PCR, that in a real time we can see if it is amplifying or not. So if you look at the fluorescence signal normally, so let's say this is your intensity of fluorescence signal. So you will see at the start it's lower, and then later on it starts increasing exponentially. So like that. So you will see this kind of a signal after each cycle, and on this side it's a time. So with the time, the fluorescence signal keep on increasing exponentially. So this curve you normally is called the PCR curves, and that you get it from during a real-time PCR, and TaqMan is one way to do that. And there is another PCR which is very similar to TaqMan, but they use slightly different type of reporter and quencher format. So it's called the Scorpion PCR. And Scorpion PCR is also one type of PCR. It works very similarly, but the only difference in this case is that in Scorpion PCR they use a DNA in a hairpin loop format. So where this is a quencher and then this is a reporter, so they are very close to one another, and then they don't give you fluorescence. But when it will open up, and when it will open up, when you see a sequence of its interest, then actually this loop will bind here and it opens up like that. Okay, when it has a target sequence. So when it opens up, then the quencher and reporter are far from one another, and then you see the amplification. So basically the point is that with the amplification, as it's getting amplified, you want to see if your product is getting amplified or not. So these are the three different standard techniques for real-time PCR. Sometimes we also have it's written that it's a real-time qPCR. There is also small q attached there in a PCR when you see in the literature, and the q stands for quantitative. So when you are also quantifying how many, let's say if you have an unknown sequence, okay, which you don't know, let's say a blood sample, we don't know how much, or serum sample, we don't know how many copies of RNA are there. It may be 1,000 copies in 1 ml, it may be 10⁶ copies per ml. And sometimes it's very important to quantify how many of those viruses are present, especially for treatment. In HIV patients, for example, if they are on ART, like therapy, or hepatitis C, if they're on therapy, then how do we know that the patient is getting improvement? And if the viral load, which means the virus concentration in blood, is falling down. So how we do it, we have to somehow quantify also this the PCR product, that we need to find out whether the starting material, although we are seeing amplification, it means the virus is there, but how much of that is there? Do you know any idea how we can really quantify that? Any thoughts?
How much DNA is in the, I know it absorbs at a certain wavelength, so there's like a machine you use, and you can get the absorbance based off that wavelength, and then you can see how much RNA or DNA you have.
So let's say we have 1,000 copies of DNA or RNA in the solution. So copies means 1,000 similar fragments, or 1,000 viruses, for example. and we start putting PCR, polymerase, and cycling, and we start seeing amplification, and then in other cases there may be 10,000 copies, right? And again, in that case, we also see the amplification. So how we can really tell that, okay, this was a 1,000, this was a 10,000. So that's where the PCR curves come into play, and there's a method to find out. I'll go back to where we were. So I showed you only one curve before, right? So let's say on the x-axis, this is the y-axis, so we have cycles. Means 1 cycle, 2 cycle, 3 cycle, 4, 5, and each cycle can be about 1 minute. Okay, each cycle means each cycle of amplification, and it continues for a long time. And then on y-axis we have intensity only, like intensity what we are getting in terms of fluorescence signal. It doesn't have any units, arbitrary units. Okay, so I showed you one curve. So one curve looks like that, right? So it start amplifying, and after some time it go to actually saturation also. And then now think about if there are less viruses present in this case.
Which are the units though?
Arbitrary units, no units. Intensity doesn't have units, like it's just intensity of light.
Yeah.
So now let's say it keeps on increasing, and we don't know how much is the concentration. Let's assume this was some X concentration. Okay, and now I put another sample, and that curves looks like that. For example, so this was X concentration. So this curve, do you think it will have a higher concentration of the virus or less concentration? It will have a higher concentration because it start increasing the intensity much quickly. So what we do is we also define some threshold, because there are some background noise also. Let's say if there is no amplification, you will still see some curves there, and that may be because of background intensity or dye itself. So we ourselves actually define some threshold based on our process. Let's say this is our threshold. Okay, this is threshold, and we consider the amplification is occurring when your signal crosses that threshold. Okay, so it means in this case it is crossing threshold here, and in this case crossing threshold here. Okay, so far it's fine. So what we can do is, now let's say if we look at what cycle it crosses the threshold. To make it simple, let's say sometimes it's between those cycles, okay? But to make it simpler, let's say this crosses threshold at cycle number 2, cycle 2, and this crosses threshold at cycle 4, for example. No, let's say 5. So from 2 to 5, so one crosses threshold at cycle 2 and one crosses threshold at cycle 5. So it means, from this, can you tell me how much times one would be more concentrated than the other one?
Because one would have 2 squared. The more concentrated one would have.
How many times more concentrated, based on this information? 2 to the power 3. Okay, so it's 3 cycles. So basically, if you look at the change in cycles, it will be 3 cycles, right? 3, right? Delta C, like change in cycle, in this case 5 minus 2 will be 3, right? And each time it gets doubled. So we can say that it's 2 to the power 3 time dilution, means time more concentrated. So that one information we get, that one is 8 times more concentrated than the other one, correct? So if we know out of these two one of the concentration, can we find out the other one? Yes, because we know the concentration factor from these curves. So in these PCR, and when we do the quantification PCR, we always run a control. So the control where we already know that it's a positive, it means it has that virus segment, and we also know that what's the concentration because we designed our control. So let's say in this case, this control was 1000 copies per ml, which we already know. So this should be 8 times more concentrated, so it would be 8,000 copies. So in that way we can also quantify the viral load. So it will not be 100% perfect, but you will get very good number in quantification. And you see these curves very often in PCR, so it's not something difficult. So they do it every day, people who run it. So there are quite a few different types of PCR, some modifications. One is, let's say, when we are interested to look for only single base pair differences, means some genes may be mutated because of a single base, and that in that case amplifying products is a little bit difficult because your control, or means your unmutated segment, will also have exactly same sequence except that one base, right? So in that case, so the both sequences are very similar. Okay, so this is a gene, this is A gene, this is a B gene. B gene is mutated, that it has only one base pair which is different. Otherwise, it's the same sequence. So if that's the case, how can you really identify that this gene has a single base pair different from the gene A, which is healthy gene? This is cancerous, let's say. How we can tell with the PCR that this gene which we are looking at right now, we don't know whether it's a cancerous or not cancerous. So because both genes are similar except one base, so if you put primers and if you amplify it, so it may amplify in both cases, right?
You would use a different color dye for each base pair, of which.
For then we'll be sequencing it, not really. Yeah, we can sequence it. Like we can sequence the whole both genes, and from sequence we'll know that okay, what is the sequence of this one and that one. In PCR we don't get a sequence. Right? In PCR, we only knows whether that specific segment is there or not, and that's why it's easy to do. Like for virus, I don't really need to spend time and money on sequencing the virus. What I can do, I can see specific segment and amplify. If it's amplifying, it means it's there or not there, and how many of them are there, right? So PCR doesn't give you a sequence of the DNA; it just amplifies some specific sequence, which you know. So how do you think that if there is a one base pair difference, we can, so basically we have to design our primers, because primers are the ones which allows the polymerase to come and attach and start elongating. So if we design our primers such that they bind to a segment where there is a one base pair difference. So if there is a one base pair difference, then they might not bind there, right? Because they will bind to the place where they are perfect complementary. So then you have to choose primers based on the area which can have that mutation. So in that way, if there is a mutation, it will amplify, not amplify based on what you really want to amplify, but you have to design primer such that if there is a one base pair different, it should not bind. And that's where the temperature also plays a very important role. Why? Let's leave it there if you don't get it, but you'll get it with the time, okay? Otherwise it will be complicated. And the second thing is that, so there are quite a variation of PCR, like different applications. The one very important is called the RTPCR, which is a reverse transcriptase PCR. And the difference in the RT-PCR and the normal PCR is that the normal PCR amplifies the DNA. In RTPCR, what we do is we first, let's say if there are RNAs, HIV virus is an RNA-based virus, Zika is also RNA-based virus. So quite a few viruses are actually RNA-based, so they are not DNA. And there are differences between DNA and RNA. Can you please tell me what are those differences?
RNA uses uracil.
RNA has a uracil instead of thymine. That's the one difference.
Yeah, DNA is missing a sugar.
No, sugar is there. DNA is DNA has a deoxy. So in a C2, it doesn't have a OH there. It only have a H there. Okay, that's the two major differences, and they're very important. So we normally convert RNA into DNA with another enzyme. That enzyme is called the reverse transcriptase enzyme, like a polymerase enzyme. So this is another enzyme which converts from RNA into complementary DNA. Okay, it makes a complementary strand of DNA. And then you have primers. The rest of the process stays the same. Once you have one complementary DNA, then you can work on the DNA. So the question is, then you can amplify it. So it means in this RTPCR, you only need another enzyme which copies information from RNA and make a complementary DNA. Complementary means opposite. So the question is, why can't we just amplify RNA, right? So why we really need to first convert it into DNA and then amplify it? So because RNA has a similar sequence, so why not we just amplify RNA? So then we can skip this step, first conversion into DNA and then amplify it. So some enzymes doesn't work very well with the RNA, and the major reason is that it's very hard to work with RNA. RNA degrade very rapidly, and that's one of the reason maybe the nature has stored our information in the form of DNA, not as RNA, inside our chromosomes. Okay, because RNA can degrade very quickly. And why RNA degrade quickly? And the answer goes by a very important question from exam point of view also. Just keep that in your mind. I can ask why RNA degrade quickly than DNA. And it goes back to that C2OH group, which is actually in a RNA. So the hydroxyl group, that can deprotonize very rapidly. So it means when you have OH, and if there is a water there, and water also have HOH, so the OH from the water can react with this OH and it can make another water molecule, and then the remaining will be just O there. So this is a more reactive group as compared to just one hydrogen. So when you have OH, the H goes away with the water, then O is only there, so it's an unsatisfied bond. So then O actually bonds with the phosphate, tries to bond with the phosphate. Then at the end of the day, it breaks that backbone, because when you phosphate trying to making a bond with your C2, which normally doesn't, then it breaks the previous bond which it has made with the C3 and C5 and makes a new bond, and then it breaks that bond also from the other side. So basically your answer, if you just put it that, is because of the OH group on the C2 carbon in a pentose sugar. There will be correct answer. I don't want you to go into more details, but I just want to give you some perception that what's chemical reaction is going on. But it's because of the OH group, and because of a reaction of OH and the OH in a solution. So now, if the OH from a solution is reacting with the OH in this case, if we store RNA at high pH, let's say pH 10, will it degrade quickly or slowly? Slowly. At high pH, we have, when we have a high pH, do we have more OH groups? We have less OH groups? We have less OH group, okay? What am I missing? So one is pH, pOH. pH 10, we have less OH. Let me think. Yeah, we have, I think what you're saying is reverse, no? At pH 1, we have more H groups, right? At pH 1. At pH 2 we have less H groups. At pH 3 we have more less H groups. At pH 7 we have neutral H and OH. And pH 8, 9 we have more less H and more OH. Right? So pH 10 we have more OH present, because OH can now react with the OH in the carbon to make water from there. So basically at high pH your RNA can degrade much quickly as compared to, okay, so it can degrade much quickly as compared to DNA, because so normally if you have RNA you don't work at a pH more than 8, because if it's more than 8 pH in a solution it can degrade very rapidly. So, and that's a part of the reason why we always want to convert RNA rapidly into complementary DNA, because once it's a complementary DNA then it is much more stable than the RNA. Okay, but so the viruses are RNA based, many of them, and then we put another enzyme actually which transfers, let's say this is a mRNA, messenger RNA for example, which we want to amplify. So with the reverse transcriptase enzyme, first it makes a complementary DNA, okay, based on the RNA sequence, and once you have a DNA, then the rest of the process will be the same, the amplification. I'll just show you one image because the time is getting over. I'll just show you one image, and then we'll stop the lecture. So this image, we'll continue with this image. Actually, I also forgot to talk about the homework. Let's talk about the homework. So homework 3, I already put it on a canvas. This is due on 4/3, which is next week, Tuesday. And the question is written there, so I think there's no need to explain. So just read the question. This is another amplification method, and the questions are asked, so just you have to answer in one page only. Okay, one page should be good enough. So time is up, I will stop there. And I will continue from here where we left. So but question, homework 3 is due next week, Tuesday.